Hello, everyone. Thank you for being here. And my name is Sona Rivas Tumanyan. I'm first a faculty member here at the School of Dental Medicine and also the leader of the Research Design and Biostatistics core at the Puerto Rico Clinical and Translational Research Consortium. And today I would like to give a brief introduction of this effort that Puerto Rico PRC-TRC Clinical and Translational Research Consortium in collaboration with the Comprehensive Cancer Center put together. And we have invited experts in human microbiome data and they want to see the speaker.
There you go.
Okay, so this is a two-day seminar series, and we have the experts invited speakers coming and presenting their research they've done in the area. And today, Dr. Amarcia Cruz will present our first speaker, Dr. Beth McCormick, but we also have a boot camp workshop that will be done tomorrow morning, and the announcement went out, and we had invited people who have prior experience in that to have a hands-on experience analyzing the microbiome data, which will be followed by a seminar on human gut microbiome and neurocognitive performance in HIV infection conducted by Dr. Josue Perez, another one of our invited speakers. Should I mention the opportunity to meet with the speakers? Please take your opportunity to meet with our speakers, and if you need a one-on-one time, you can contact our Zapti, the one who sent out the announcement, by email or by phone, or pass by her desk at PRC-TRC to get your name on the list, pick up time if you need one-on-one time with our experts. And thank you for being here, and I would like to leave the podium to Dr. Amarcia Cruz, who is the head of the PI, one of the PIs of PRC-TRC, and also the Comprehensive Cancer Center. Thank you for bringing us the opportunity to hear from the best. Thank you.
Let me have this. You have it there? Thank you. Muy bien. Well, good morning to everyone, and good afternoon already, and thank you for being here. We're extremely excited to have the opportunity to have this seminar transmitted via webinar to our partners in the Ponce Health Sciences University and the University Central del Caribe, which are the partners for the PRC-TRC. And I'm extremely happy to be able to present to you this first lecture as part of the two-day workshop that Sona helped coordinate as part of the PRC-TRC core. And our first speaker today is Dr. Beth McCormick. She did her bachelor's at the University of New Hampshire. Then she did a PhD at the University of Rhode Island. And then she had two postdoctoral experiences, both at Harvard Medical School, one in cell biology and the second one in gastrointestinal pathology. She is a professor at the University of Massachusetts Medical School and the vice chair of the Department of Microbiology and Physiological System at the same medical school. She has been funded by the NIH, by multiple different institutes for many years. And just to mention some of the topics that her group have been leaders on, include salmonella pathogenesis and processing of secreted effectors by caspase-3. She also has the University of Massachusetts Center for Clinical and Translational Science, and I think that's one of the key factors why we were able to collaborate with her, because as a basic scientist, she works with people that are from the clinical field, mostly in the field of inflammatory bowel disease, infectious diseases, and also cancer. Today we were talking briefly, and Beth told me that she's an equal opportunity, and I said, I like that. You know, so it goes across the spectrum, and in fact today and tomorrow we're also going to be looking at the microbiome from different organ models, so including cancer, but also talking about HIV, and Josue will be talking about that tomorrow. So it's going beyond the gut, if we were going to call this, right? As a gastroenterologist, of course, I think about the gut all the time, but never thought that those organisms that are in the bowel really modulate so many of the functions that are related to diseases, much farther from the gut that no one thought originally. Dr. McCormick has also multiple papers in peer-reviewed journals, and it's one of the qualities that distinguish her. It's being a mentor. And we have been touched by that. And in fact, she is part of our grant. We obtained a grant to examine some of these markers in the gut from patients from Puerto Rico. And so we're really grateful that she has allowed us to continue to expand our knowledge in that area that is so novel this day. But as she told me today, this has been, she's been working on this for over 20 years. So, new for us, but, has been there for a long time. Without any further ado, I would like to welcome Dr. Beth McCormick as the first speaker for the two-day seminar series. Beth.
All right, so thank you very much. Can you all hear me? One of the biggest challenges is to be tall enough so I can see things, but yet, all right. So just let me know if you can't hear me, and I would be happy to speak louder. So I want to first start off by thanking the team here for inviting us down. It's always a pleasure to visit new places, and if you can visit a wonderful place like this and foster a partnership, that's even better. So I'm going to be talking today about our efforts in the realm of studying host microbial interactions and identifying new insights underlying the mechanisms of mucosal inflammation. And so my first challenge is to advance the slide, which I cannot do successfully here. Oops. I'd like to be able to do it more manually than. Okay, so we solved the secret code of how to advance the slides. All right, so I'm going to be focusing today's lecture zoning in on the intestinal mucosa. I don't think this needs a large introduction by the community sitting here. But I just wanted to point out that your intestinal tract is really nothing more than a single layer of epithelium that on one side has this hundreds of trillions of microbes sitting in the external milieu. Whoops, now I'm advancing it, whoops. Jane, stop this crazy thing. Okay. And then on the inside, you have this really sophisticated, coordinated, innate and adaptive immune system. And so if you take a look at this by cartoon and schematically, this is what you see, is you have these commensal communities out here in the lumen that confer education of the immune system sitting underneath here via signals by the dendritic cells that then confer signals onto your T regulatory cells, and in this beautiful coordinated manner, keep a homeostatic flow to your health and well-being. But when you have perturbations in this ecosystem, primarily looking at here with an infection with a microbial pathogen, the intestinal epithelium has to be significantly poised to interact appropriately and immediately. And so then you see here that you have this pattern of signaling molecules being elicited in these secreted inflammatory mediators, primarily of these cytokines and chemokines that then allow your first responders in this immune system to start to appear. And you can see this here by the introduction of these neutrophils. And so this signifies that these, when you have a more marked inflammatory response, that these neutrophils are well poised then to act as first responders. They come to the site of infection. And you can see here that they're nothing more than bags of enzymes, or they once originally thought were to be these bags of enzymes that are deployed. They have reactive oxygen species, lots of AMPs, chemokines, and these nets. So they really do this holistic kind of deployment of their collateral in terms of recognizing and then deploying their arsenal to get rid of the pathogen. But the problem is that in doing so, you can see here that there can be a lot of collateral damage. So the network and the fine-tuning of allowing neutrophils come in to sort of maintain and forego the infection needs to be balanced with diseases that go awry. And so when neutrophils are good, so then in a successful response where you see neutrophil infiltration is then followed by apoptosis and then secretion of pro-resolution factors that restore the intestine to a homeostatic state. So you can see this here, the neutrophils are then attacking the organism, but you see these feedback mechanisms that also can react on the tissue as well. So when neutrophils go bad, such as inflammatory pathology seen here, it can be really problematic. So neutrophil infiltration is characteristic of a whole host of diseases, including inflammatory bowel diseases such as Crohn's disease and ulcerative colitis. This is mimicked in infection with inflammatory pathogens, as you'll be seeing with Salmonella, for example, and Shigella. They promote transmural inflammation, they cause crypt abscesses, and there's also, because of this leakiness, with their ability to cross the intestinal epithelial barrier, you see also increased epithelial permeability that also translates with bacterial translocation, which then can amplify an inflammatory event. So this process of recruiting neutrophils really has to be fine-tuned because, as you can see here, when it goes awry, it could lead to some significant pathologies. And so my work in my lab for the many decades now is to try to understand the factors then that control neutrophil infiltration into the intestine during health and disease. And we are interested in this for three primary reasons. So this is sort of what I tell the grant reviewers when I write my grants. This is why we're interested in studying this, is because neutrophil transepithelial migration is a rate-limiting step of crypt abscess formation. So if you take a look over here in this cartoon, it really depicts a pathogen coming in, and then you see this underlying inflammatory response with the neutrophils coming out of the blood vessels, then crossing the laminar propria area, and then subsequently and finally crossing the mucosal barrier. And so what we're going to be talking today mostly is this identified here in this box, and this is the process of neutrophil transepithelial migration. And so, oops, sorry. Shoot, punchline. This pad is really sensitive, so I apologize. So anyway, so our understanding of how neutrophils cross this mucosal barrier is significant because it's the rate-limiting step to crypt abscess formation I just mentioned. Also, the numbers of neutrophils that transmigrate across the mucosal barrier correlate with disease severity, and it's also the key event in mediating the clinical manifestations of both infectious as well as idiopathic gastroenteritis, such as IBD. So this is where my story really begins, is the eating of your grandmother's chocolate chip cookie batter, which may be okay, but in certain instances, this is what may happen to you. So this is the consequences of dipping into to undercooked eggs. And so Salmonella is an organism that causes salmonellosis, and is hallmarked here by really robust GI distress in terms of abdominal pain, cramping, diarrhea, vomiting. It is a febrile disease. It is self-limiting, so it's a nasty disease lasting 3 to 5 days. If you're healthy, if you're immunocompromised, it can be more severe, and in certain cases can cause death. 25% of patients infected with Salmonella head to the hospital, and we're seeing a more robust multi-drug resistance phenotype of late. 20% of those isolated strains of Salmonella are multi-drug resistance isolates. So in order for Salmonella to become pathogenic, it utilizes a type 3 secretion system. And for those of you that don't really know a lot about this, this is a really interesting, sophisticated system that allows effector proteins that are generated inside the Salmonella bacterium to be effluxed through a syringe-like or a needle-like structure into the host. This is predominantly from gram-negative bacteria. It has been thought to be evolved from the flagellate basal body, and it regulates then the secretion and translocation of these effectors. And these become important mediators in allowing Salmonella to not only enter into the host organism, into the host, but also to introduce pro-inflammatory phenotypes. And I've just outlined a couple of the important mediators here of these effector proteins, and we're going to hear an awful lot about one of these today called CYP-A. So this is just an electron micrograph that I actually took when I was a postdoc, in fact. And it shows you that Salmonella invade through a process called bacteria-mediated endocytosis. And in causing disease, they preferentially like to invade the distal ileum of the intestine, but they don't exclusively colonize that. And so just hallmark features of the invasion process shown here. Salmonella come in contact with the epithelium. They induce what are known as these membrane blebs that really are these really fascinating ruffles that are very important and act in remodeling. It takes up the bacteria in these membrane vacuoles, and the bacteria then are transported to the basolateral milieu, or it's taken up with the reticuloendothelial system, making its way toward the spleen and the liver. This is also partnered with their ability to induce active states of intestinal inflammation. And this is from a patient who did not have a great outcome with this disease. As you can see here, a hallmark pathology of the disease is much like what you see in patients with ulcerative colitis. You see these areas of ulceration. And if you were to blow that up in terms of histopathology, you see a bunch of neutrophils marching across the intestinal epithelium, forming these crypt abscesses. I always like to sort of tell a little bit of history of how I got this particular slide. So when I was a postdoc fellow at the Brigham and Women's Hospital working on this, I got approached by the leading GI pathologist in our group saying, I have this slide for you. And he was really remarkable in terms of his observation, because this is exactly the pathology that you see in patients with ulcerative colitis. And they actually thought this patient had ulcerative colitis until they did a positive stool culture, and the patient actually had salmonellosis. So this is Jim Crawford. He's now down in Florida. But I was also struck by the little storyline here. This, group of GI specialists thought that he had UC, and he actually had salmonella. So we are able then to model, and we'll go back to the significance of that statement in a little bit. So when I was a postdoc, I set up a model system in which we could try to figure out mechanistically how salmonella was driving this inflammatory event. And so this just shows this model system here. So we have these transwells, and we're able to grow on these transwells a layer of TD4 cells, and I can hope you can appreciate that we're growing them in the inverted fashion here. So the apical aspect is pointed down here. The basolateral aspect is actually attached to the filter, and this is because we like to go with gravity. We then will isolate human peripheral blood neutrophils, put that in the upper chamber here. So we have things that are all nicely physiological oriented. You have salmonella coming in contact with the apical surface, and then neutrophils interacting from the basolateral space. So the question was, can salmonella epithelial interactions drive subsequent neutrophil migration? And this just shows you our action slide, if you will, of neutrophils actually migrating in this context. And the answer is yes. And so we spent, I do that, the collection of we. I have spent then the next remainder of my career trying to figure out exactly the mechanisms that underlie this response. And so this sort of is a snapshot of our inroads into modeling of how salmonella induces mucosal inflammation. And so you have salmonella coming in, interacting with the intestinal epithelial surface. You get deployment of a couple of these effector proteins, SOP-E and SOP-B, that then drive signaling transduction cascades, which leads to the activation of MAP kinases, which then upregulate NF-kappa B. You then also get the deployment of the flagellin transcellularly across the epithelium, where it interacts with TLR5. Both of these signaling pathways, the MAP kinase pathway, in addition to activation of TLR5, educate NF-kappa B to to engage pro-inflammatory molecules such as IL-8, which are released to the basolateral aspect down here. Recruiting then neutrophils out of the bloodstream and into the subepithelial position. I think we are also in the field led to believe that IL-8 does everything. I'm here to tell you it doesn't, okay? So that's my disclaimer. It's really critical in getting neutrophils from out of the bloodstream, but there's secondary mediators that permit neutrophils to advance beyond the subepithelium, and that are shown here. So in addition to these other effector proteins, we know that there's another one called CYP-A. CYP-A turns on a very important arachidonic acid cascade involving 12-lipoxygenase pathway. The 12-LO pathway leads to the synthesis of a very potent neutrophil chemoattractant called hypoxylin A3. Hypoxylin A3 is vectorially released to the apical surface by an ABC transporter called MRP2. And you can see here that then you're going to form a gradient of hypoxylin across the tight junction barrier, leading then for the neutrophils to migrate out and go to the site of infection. And so for the next few slides, I'm going to focus on MRP2 and its importance in this pathology. So MRP2 is an ATP binding cassette transporter. I think we all know it to contribute to drug resistance, and there's two flavors of these ABC transporters, P-glycoprotein, which is encoded for by MDR1, and there's also a host of multi-drug resistance associated family member proteins, MRP1 through 9. So these are evolved in us and are involved in the excretion of toxins from cells. So these are our garbage removal mechanisms that we have, and we need to have them because if we don't have them, we're going to die of our own sort of toxins that we get from the environment. So they eliminate xenobiotics. But there's been this really fascinating kind of undercurrent of literature that's been coming out in the last 10 years ago to suggest that these ABC transporters also mediate processes of inflammation. And there's a couple of examples that were in the literature when we started out. That LTC4, which is leukotriene, utilizes MRP2 pathway for its efflux. And then there's been some anecdotal observations to suggest that they also play a role in host defense in both the lung and the gut. The distribution of these transporters in each tissue is going to be specific for the tissue. In the intestinal tract, we see a large preference of many of these transporters being on the basolateral surface, although we do see a couple on the apical surface, MRP2 being one of them. And so when you look at infection with S. typhimurium, and this is an in vivo model system of S. typhimurium, this is a control, so this is our mock control where we feed buffer to the mice. And then this is in the presence of wild-type salmonella and a CYP-A mutant. You can see here that in the presence of wild-type infection that there is this beautiful upregulation of MRP2, and this is specifically localized to the apical surface. And here in the absence of CYP-A, you do not see such upregulation. We can bring this reductionistically and look at what happens so this is in tissue culture, when we've made an MRP2 mutant shown here in blue versus control and an irrelevant transporter knockdown. And this is our question of, so what happens when you drive salmonella infection and look for PMN transmigration? We can see here the importance. So if MRP2 is important in effluxing out hypoxylin A3, when you downregulate that transporter, you lose hypoxylin efflux, you lose the ability of salmonella to induce neutrophil transepithelial migration. And this was also correlated with the inability of this MRP2 knockdown to secrete hypoxylin A3. So all of this makes sense, right? So you get this inflammation, you get an uptick in MRP2. If you prevent that, so if you knock out MRP2, you don't see the secretion of hypoxylin and you don't see neutrophil transmigration. So we have coined this as the hypoxylin MRP2 axis. This is a new innate immune pathway that you probably never heard about, and so I'm telling you about it today. It's really critical in underlying the control of neutrophils across the mucosal epithelial surface. And so we've taken a look at this in a variety of different diseases, and we are beginning to understand that this important axis is a conserved pathway. So I've told you about our efforts with Salmonella. This is also mimicked in infections with Shigella and also other enterics such as Enteroagave coli. This is even modeled with that German outbreak strain of a couple years ago. This is not specific to the GI tract. When we looked at other organs, particularly with respiratory tract diseases, this is also modeled in diseases induced with Pseudomonas aeruginosa, Klebsiella pneumonia, Streptococcus pneumonia, and M. tuberculosis. So I have all of the gram-negatives covered, I have gram-positives, and I even have an acid-fast organism in there. They all kind of fundamentally engage this pathway for this innate immune pathway. Whoops. And so if you take a look at this evidence in terms of most of these, I think you can argue are acute diseases, perhaps save for M. tuberculosis. And so when we started to take our observations to the literature, I was struck by a couple of different studies in terms of the observation of hypoxin A3 in more chronic inflammatory diseases. And this study right here represents a study with Lyme's arthritis induced by the organism Borrelia burgdorferi, causes Lyme's disease. And hypoxin, in terms of an inflammatory mediator, was the most highly and abundant at the site of this inflammation event. We have not proceeded with studies with looking at this, but it's, I think it just begs the question of how broadly this axis is deployed. And if you take a look at what was been done with psoriasis, so this is the first non-infectious disease in which they saw hypoxilin in humans under a pathological situation. And so we feel that this is going to be a very important inflammatory event that we can hopefully intercede with some interesting therapeutics. And I'm going to talk about our efforts on that in a second. So I told you that when I grew up in the GI world, I was a postdoc fellow, and I had this observation with Salmonella, and I became keenly aware that the observations in the disease pathogenesis of Salmonella really mimics patients with ulcerative colitis in terms of clinically, radiologically, and histopathologically. So when I had this result coming from the table, looking at Salmonella, I thought, is there any sort of correlation between what we're finding with Salmonella pathogenesis and more idiopathic diseases such as Crohn's disease and ulcerative colitis? So we took a look at what happened in patients with IBD. And basically, this is healthy intestine, and this is Crohn's disease patients and an ulcerative colitis patient. And this shows you here the expression of MRP2 on the apical surface. Okay? And so now we're starting to probe the importance of that, because if we think about this in terms of modeling it, in terms of intervention, and if we want to sort of limit the numbers of neutrophils that are coming across, could we then think about targeting MRP2 in terms of patient therapy? And so this is the larger question. Does the MRP2 hypoxin A3 axis drive non-infection inflammation? And so to do this, we decided to go back to our model systems, and we use a DSS colitis model. This is a model of acute mucosal injury, inflammation, and recovery. So basically, we put mice on 3% DSS in water, and over time, what you'll see here, you have this acute phase of inflammation. In about 10 to 12 days, it peaks. Oops, shoot. And then we see this more, a resolution probably after 2 weeks. And this is why we love this model, because we can see, this is, so, Marcia could probably agree with me, that this is the most well-utilized model to look at colitis, but yet the most maligned. And we like it because in a very short period of time, you can see a disease progression with resolution. So you can think of studies that you can intervene in terms of seeing if you see improvement, and then if anything impacts the resolution phase. And so this just shows you the histopathology that are shown at day 0 versus day 10. So you can see here it's really an inflammatory-based disease model for us. And so what we did was, once we got those animals on DSS at their acute phase of inflammation, we took the proximal and sort of the mid-colon area here. We opened them up, we got mucosal scrapings, and identified that in disease treatment here with DSS that we see a large a robust amount of Hopoxin A3, all right? This is a first demonstration in the GI tract in vivo that this has been demonstrated, so we're very happy to see that. And so then we thought, okay, so our premise was if we block MRP2, we're going to see less Hopoxin A3, and then hopefully lessen the ability to induce neutrophil transmigration. And so what we did was, to test that, we then coupled probenecid with a conjugate. I'm not allowed to tell you what that conjugate is because it's under IP issues right now. But the essence was to make probenecid, which is a normal inhibitor of MRP2, non-permeable. We didn't want it to become systemic, and we wanted to be able to target it more topically. And so then we treated the animals with probenecid. So this is, again, looking in your DSS model, coupled with probenecid treatment. And you see here, if you take a look at the histopathology score, this is vehicle controlled, this is the drug treatment, that there was a decline in the disease severity. This is shown here. Histopathologically, where you see a less accumulation of the neutrophils coming into the tissue. And this was also coupled with an increase in the length of the colon. So when you have disease in these animals, what you see is shrinkage of the colon, and when you see improvement with health, that the colon length increases, and this is exactly what we find right here. But this was not so while we saw a decrease in our disease severity index, this does not mean that we had wholesale tissue elimination from the tissue. So shown here very simply is that tissue immune cell infiltration is unaffected by blocking Hopoxin A3 secretion through MRP2. So this is our total CD45 plus population vehicle with the probenecid conjugate. And this is the percent PMN among the LPLs. So very, very solid numbers. So we're not affecting host cell immune function there. But interestingly, we are specifically affecting the neutrophils to migrate across the intestinal epithelium, because when you take a look at tissue neutrophils compared to neutrophils in the feces, you see here that in the presence of the drug, we have a downshift of the ability of neutrophils to migrate across the epithelium. Right? So this was not a one-trick pony. We were able to demonstrate this in more immune-driven models of colitis, and this is the CD45 RB high model. So basically what happens, you take collect the CD45 RB high population of T cells, put them in a SCID mouse, and what happens over a period of 6 to 8 weeks, they will develop a very human-like colitis. And with the progression, as you can see here, there's very pronounced structural prolapsing, which results in about 70% of these animals. And so what we do here is we get the animals sick. So it takes about 6 to 8 weeks to start to see these animals go into a decline. And we do this by measuring weight and just looking at their fur phenotype. And as soon as we start to see them get sick, we then therapeutically intervene with the probenecid conjugate. And we basically don't, so these mice here, if you were to sack them, you would see that they have this really disease severity index is high. We have done rectal, on these animals that have had rectal prolapse in them. So we delivered this by enema. And we have returned rectal prolapsing to health. But these bums, I hope you see here, if you compare this bum to that bum, that's different, right? Even this bum to that bum. So we can reverse disease in these animals by treating these animals with the probenecid conjugate. So on the strength of these data, we are wondering whether or not we can use this information with Hopoxin MRP2 regulation as a model of therapeutic intervention for patients with IBD. So with my longtime collaborator and scientific partner, Andy Mersony, we have designed a drug targeted to MRP2. And so we hope that this is the concept, that if we prevent transient disruption of the epithelial barrier caused by the neutrophils and reduce crypt abscess formation, and you're likely overall to have, less likely overall to have intestinal dysfunction. We are now in phase 2 clinical trials, focusing on ulcerative colitis patients, and this is done with our partners at St. Bartholomew's Hospital and the London's Hospital in London. So stay tuned for that. So that sort of is a summary of the pro-inflammatory aspects of what we've been working on. But there's balance in the intestinal tract, right? So if you have a pro-inflammatory response, there must be something in your system that sort of tries to compensate for that. And you can see this here throughout, our immune system, where you have your innate defenses at a battle with your constant immune surveillance system. You then have tolerance to commensal bacteria, which is balanced by infection with pathogenic bacteria. And then you have regulatory immune responses that have to oppose these strong inflammatory responses. So on the tenet and thesis of that, we wondered whether or not there was a factor that balances out the hypoxia-mediated inflammation. So I told you about this pro-inflammatory arm that's mediated by the MRP hypoxin axis, but is there something that inhibits transmigration? And again, taking a dive off of a very steep cliff, Randy and I began to wonder about that. And so we surmised that there might be a factor that is released through another transporter that then works to inhibit transmigration, so to balance this out. And this crazy notion had some rationale to it. I had published an earlier paper looking at that salmonella does functionally downregulate P-glycoprotein, which is encoded for by MDR1. And now we found that PGP is restricted to the apical surface of polarized epithelial cells and then is a determinant of homeostatic interactions between bacteria and the human host. When we had this data, and this just shows that when you infect a mouse with wild-type salmonella, you see wholesale loss of protein expression in P-glycoprotein. But when you have infection with a mutant that does not express the type 3 secretion system, you see a response that mimics the uninfected control. And so when I had this data and didn't really know much about P-glycoprotein, I began to sort of read the literature. And at the same time that we were making our observations that infection with salmonella downregulates P-glycoprotein, there was this really nice body of literature starting to emerge to also show that P-glycoprotein and its role in intestinal inflammation. And that is shown here. So you can see here that healthy individuals have this robust expression of P-glycoprotein, but in individuals with an inflamed intestine, such as those patients with ulcerative colitis, you see a collapse of the expression of P-glycoprotein. So P-glycoprotein expression is strongly decreased in inflamed intestinal epithelia patients with intestinal GI disorders, and even in knockout mice that have MDR1 knockouts, they experience ulcerative colitis-like symptoms, and that now there's an understanding of a single nucleotide polymorphism in the MDR1 gene, which correlates with lower intestinal P-glycoprotein expression and higher incidence of UC. Okay? We do not think that these observations that we made with salmonella, with these observations in ulcerative colitis, is just a coincidence. And I'll tell you why. So we started out with our hypothesis that P-glycoprotein is going to be critical in releasing a factor that we call amend. At some points in times, I used to call it amen, but we want it to. And so this is activity modulating epithelial neutrophil discourse, all right? And so, which is the yin to hypoxin's yang. So we think that there is a receptor sitting on the neutrophils that is sensitive to this factor that then dampens the control of neutrophils and wants to shut it down and impede the migration. So how we went about doing this is that we went back to our TD4 cell model on filters, and this time you're going to see in the more conventional manner with the apical surface facing up at you. And so we did a control where we have, this is just the normal TD4 cells, and then we made P-glycoprotein knockdowns in these cells as well. We collected the apical supernatants, we concentrated it, and then we applied it to our monolayers with an imposed gradient of hypoxin A3 to see if this factor, we could isolate out a factor that then stopped hypoxylin-driven neutrophil migration. And the answer is we found this factor. So this takes a look at what you see here, and all this is normalized. So this is neutrophil migration normalized. So this is our result with hypoxylin. When we collect out from the control TD4 cells, we do isolate a bioactivity which suppresses the ability of neutrophils to migrate to hypoxylin. But you can see here, we did a couple of different knockouts of P-glycoprotein, that the suppression is released. So we're not getting that factor being released, and therefore if that factor is not released, we're not dampening PMN transmigration. So again, we refer to this as amend here. We were able to duplicate this in mice. Remember, we have mice with a phenotype with a P-glycoprotein knockout. And so going back to our mucosal scrapings, we get our mucosal scrapings, and we isolate that factor. And so in wild-type mouse extracts, this is hypoxylin, we isolate that out. We can see here that there is a factor that immunosuppresses PMN-driven migration, that as we dilute this out, we dilute out the effect. But in mice in which we have that P-glycoprotein knockout, we don't collect such a factor, okay? So this is very, you know, our in vitro findings led us to have this similar phenotype in vivo. So what the heck is amend? So we had the fortunate opportunity, right, that we can collect amend in the absence of and presence of a P-glycoprotein inhibitor. And so it went through an HPLC separation. We isolated fractions, and then we did the amend bioactivity, and we went back to our salmonella-induced PMN transmigration assays. So as you can see here, this is very low throughput. So this poor postdoc fellow that I had working on this went through thousands of fractions before we found the eureka moment. And so the upper tracing here is untreated, and then the lower tracing here is P-glycoprotein inhibition. If you zone in right here, you see this blip, a peak here in the untreated that is absent in the P-glycoprotein inhibitor cohort, right? So this is what we zoned in on and went after. We identified it by HPLC and then mass spectrometry. Shown here is the line here, the scramble control, and here is the diminished line in the P-glycoprotein knockout. Okay, so this is the big reveal. So it happens to be a family member of the endocannabinoid class of molecules. I know, see, this is what you're all thinking. I'm sure that this is where you're all going here, but it's not the case, because these are natural products that have a completely different structure, folks, and that are non-psychotropic and that hit off a different receptor. But believe it or not, we have these endocannabinoids that are floating through our bodies as we are currently sitting here taking down notes that Dr. McCormick is such a great speaker and does great work. Right, so, but look at the amount of functions that they are involved in. I mean, our memory, our ability to ward off certain diseases such as cancer, and they control our appetite, our fertility, cardiovascular diseases that are all connected in a way in which I never appreciated up until now. So the endocannabinoid system is very complicated. I'm going to distill it down to one slide. And so this is just an overview. So you have these endocannabinoid ligands, which are that these are important because you can see here they need to be produced on demand, they need to act locally, they are inactivated rapidly, and they bind to transmembrane G-coupled protein receptors. And so in principally inhibiting neurotransmitted release, okay? The receptors come in two flavors, CB1 and CB2. CB1 is probably the most widespread CB receptor. These are on your peripheral tissues, your brain. This is what gives you the psychotropic impact of THC. Your other endocannabinoid receptors here are CB2, which are on immune cells, okay? Yes, immune cells have endocannabinoid receptors on them. We're going to talk about that moving forward here, okay? There are two major classes of endocannabinoid signaling molecules that we release. One belongs to this N-acyl ethanolamide family, which is shown here. The end products primarily is by this molecule called AEA, or anandamide, and a couple of others called PEA. The other class is synthesized through these monoacyl glycerols, and the poster child for this is 2-acylglycerol. Okay? This is where the complexity kind of starts to creep in. One of the areas of complexity is that even though you have sort of different molecules being released through a couple of different synthetic pathways, they can cross-bind to any sort of all these receptors. Okay? And then you have the added outlier of interactions with other entourage lipids. Okay? I'm not going to go any deeper into the woods than this. I alert you to this just because this is very complicated science that we sort of stepped our foot into. But this was one of the really amazing kind of observations that we had with this. Even though you have these two major classes of endocannabinoids, one which are metabolized by fatty acid amide hydrolases, and one which is metabolized by monoacyl glyceride lipases, they both will be metabolized to arachidonic acid. Okay? Remember, if we go back to figure out where hypoxylin started from, it is synthesized by arachidonic acid. So these pathways are reciprocally intertwined. So arachidonic acid is a degradation product that feeds back into the hypoxylin synthetic pathway. Okay? So we still didn't know what molecule we were dealing with. So this was the big epiphany moment. We're working on trying to figure out how this is all regulated and coordinated, but we didn't really know what one of these molecules was the key factor. But we had enzymes that we can perturb. So that's exactly what we did. So to our amend fraction, we added inhibitors of FAAH and inhibitors of MAG-L. And you can see here that when you inhibit the FAAH pathway, you release the suppression of the factor. And this was shown here in vitro cell supernatants, and going back to our mucosal scrapings, we showed the same thing. So it turns out, in fact, that FAH is the key. And so the main product released from that, we think, is anandamide. So we had the enzymatic way of doing it, and then we also had the mass spec way of doing it, where we could pair our endocannabinoids being shuttled out from control versus P-glycoprotein. And then we can see which ones weren't being shuttled out of P-glycoprotein, and lo and behold, sitting on top is our AEA. Okay? It turns out that 2-AG is not really a substrate for this particular transporter much. So we're putting sort of all of our kind of eggs in one basket, that these top three hits of AEA, oleol ethanolamine, and virotamine shoots are it. And so we've been able now to commercially take these substrates, put them into our assay, and sort of see whether or not they will reduce hypoxylin-driven neutrophil transmigration, and they do. So this is our response to hypoxylin. This is our response with these commercially derived molecules. This was a pain in the neck to get going, folks, because of solubility issues and concentration issues. And but anyway, so we got it going. And you can see here that we tested a whole host of others that I won't alert you to in terms of what the names are, but you can go search this out if you want to. I do want to make the point, though, we saw a hit with this alpha LNEA, but not with the gamma LNEA. So this is alpha, and this is gamma. Look how closely in structure they are, but yet one we have a bioactivity from, and the other one that we don't, okay? So right now, we know that we are dealing with a specific class of these N-acyl ethanolamides, which is pretty exciting. And what we're currently doing now is seeing how important this amend pathway is in vivo. So we're going back to our DSS model of colitis and taking a look at what happens here. And so remember that the DSS model I showed you before has this really nice histopathology for colitis, showing progressive epithelial destruction. We can assess bacterial translocation, and it has this really nice hallmark of neutrophil infiltration. So we can use mice as our experimental tool because we can get CB2 knockout mice. So if you don't have the receptor to respond to amend, you're not going to, so you don't have that signaling, so they can't respond to amend because they don't have the receptor. And conversely, we can use the P-glycoprotein knockout or even the drug-treated mice. In this fact, they cannot secrete amend. So we have one model system in which they don't secrete amend, and then we have another model system where they can't respond to amend. All right, so we're coming at it from a couple of different angles. So here's our data. So we have DSS coupled now in mice with a CB2 knockout. And so the prediction is that you should see a worsening of the DSS colitis. So this is the % weight drop over time, wild type, and this is the CB2 knockout mice. And you can see here that there is this, kinetically, there is an increase in the progression of weight loss. If you take a look at the histopathological score, you see here that there's an uptick in severity of disease in the CB2 knockout mice, which we predicted. And we saw the same thing with a lack of amend secretion through P-glycoprotein worsens DSS colitis. So these are the P-glycoprotein knockout animals, again showing a quicker kinetic of weight loss that was paired with an uptick in their disease severity over time. Okay. So, what we stumbled upon, interestingly, sitting in your mucosa first, and I'm gonna talked about the intestinal epithelium. You have these ABC transporters that have been historically linked to a very important physiology in terms of being a garbage dump, basically. But what is happening is that these transporters are sentinels to kind of gauge homeostasis in sort of disease. So you can see here in the basal state, we had this really nice baseline expression of pre-glycoprotein, but very little MRP2. So this baseline state is allowing then this anti-inflammatory mediators to be pumped out, controlling, making your intestinal epithelium sort of a state of immunosuppression, which is what you want. In a sea of trillions of organisms, you don't want to be having this rampant inflammatory response. But in a coordinated way, when you have a pro-inflammatory event, even fostered by infectious or non-infectious mediators, you see an opposite happen. You see a loss of P-glycoprotein, but an uptick in MRP2, which is then releasing an immunomodulator driving neutrophil infiltration. Okay? So how is this amend pathway being regulated? So the first thing I thought of, could it be what these things are probably sensing? And that's your microbiota, right? So these things are in constant sort of synergy with what's happening with your intestinal epithelium, with your outside world. And so we know that the importance of the microbiota along a variety of spectrums, including a protective function, structural functions, metabolic functions that include our ability to resist pathogens, help us with nutrient absorption and competition. They educate our immune system. They provide a barrier for us. They produce vitamins for us. So they're critical in sort of our maintenance and welfare, right? So we wondered whether or not the commensal bacteria regulate the amend pathway. And so our supposition to support this is based on a couple of different observations. One is that these commensal bacteria are critical in maintenance of intestinal homeostasis and development, that antibiotic-treated mice are more vulnerable to DSS. Salmonella infects cells more efficiently when they lack P-glycoprotein, and they are less able to infect cells overexpressing P-glycoprotein. I left out that point. So if we were to overexpress P-glycoprotein in intestinal epithelial cells, we prevent salmonella from invading, okay? And then the final observation, streptomycin pretreatment sensitizes mice to salmonella infection. So it seems to be more complex than simple colonization. So based on these observations, we hypothesized then that commensal bacteria maintain P-glycoprotein expression in the homeostatic intestine, which is what we find. So when you deplete the microbiome, and we did this a couple of different ways. We did streptomycin treatment. This is 4 mg over a week, I think. We'd thrown in a couple of other antibiotics as well. And then we looked at the expression of P-glycoprotein and MRP2 in the intestinal epithelium. So you see here, you see loss of P-glycoprotein. You see a little rise in MRP2. This is the densitometry showing exactly what's happening in terms of P-glycoprotein being downregulated and MRP2 slightly being upregulated, supporting our model of what I just showed you, that there is this imbalance in what's happening in terms of being able to maintain homeostasis. So you're shifting the balance from the homeostatic arm to the pro-inflammatory arm. So we also then were able to show that P-glycoprotein expression and endocannabinoid secretion are reduced in dysbiosis. So again, showing here, I expanded out the view here. So this is showing now proximal, mid, and distal. So you see here that there's, there may be some sort of specification to the proximal and mid colon in terms of specificity of downregulating P-glycoprotein in terms of dysbiosis. This shows the mRNA analysis shown here, that by gene expression we also see this downregulation of P-glycoprotein happening. But look what happens when we look at what's going on with endocannabinoid secretion. So in these mucosal scrapings that we have in this dysbiotic mice, it's probably too small to see down here, but you can see here that in the streptomycin-treated mice and the cephalosporin-treated mice, that there is, if you take a look at AEA, OEA, and the virotamine, that there is a wholesale downregulation in the endocannabinoids that are being released in these animals. So this brings us back to my last slide. So we have uncovered this really interesting coordinate regulation of ABC transporters during inflammation. That the maintenance of really driving the homeostatic arm is influenced by the commensal bacteria. And when that is thrown out of whack, either by an inflammatory insult or a pathogen insult, that you see a loss of this pathway in favor of this pro-inflammatory arm. And the significance of this is that they are metabolically linked. These two pathways, which are so sensitive in maintaining a neutrophil balance, are metabolically linked by arachidonic acid. That's my last slide. This is the most important slide, however. So this is the wind beneath my wings for sure. This is my talented group of people. A compilation of graduate students, postdocs, but their collective talents are matched by none. Rose Zabadi is a talented postdoctoral fellow in my group that just received the CCFA Career Development Award. Rose is right here. She almost single-handedly has pulled the immense story, carried that on her back. So we are hopefully going to get a very nice publication out of that. It's under resubmission currently. I cannot thank enough all of my different funding sources throughout the years. And you're probably noticing this, right? I mean, how many people notice that, right? You got to raise your hand, because who gets funded by a beer company? But it turns out that you have a Danish student, Eric, which is interested in studying enteric pathogens. There is the Carlsberg group will fund you. So you have to have that bandwidth of being enteric pathogen and being Danish. So I was fortunate enough to kind of have that. And then my long-standing collaborator and good friend, Randy Mercenary, who is at the University of Bath. So I will end there, take any questions you have. And I'm thinking that those of you who are off campus who want to write in their Q&As, you can certainly do that. But I'm happy to take any questions that you have. Yes?
I think this is tremendously interesting. The way you show it, there's a possibility that you don't have P-glycoprotein, you get accumulation of anandamide and arachidonic acid inside the cell. And that may shift the equilibrium, the metabolic equilibrium towards the oxidant.
That's true.
So if you get accumulation of arachidonic acid, that's what happens. But the structure of your inhibitors, one of them is an omega-3 fatty acid. It's actually the one that's active. So it comes from an omega-3, so the double bond is positioned in the omega-3 position. The question is, what would happen is if instead of accumulating arachidonic acid, you would be accumulating eicosapentaenoic acid or
Yeah, so that's a great question, and something, so we've only been able to really crack the egg. I don't know if you've ever played around with endocannabinoids. Have you ever tried to isolate and play with endocannabinoids there? No, don't do it, okay? That's my advice, is just don't do it. So now that I think we are proficient on at least knowing what we're doing, that's exactly what we're going to be looking at, because we don't think that in our wildest imaginations that these things are working in isolation. So I think there's probably going to be more either these good, guys hanging around to help with the immunosuppression state. So we are taking a look at that. We have shown exactly that. So in these P-glycoprotein knockout cells, we've been able to show that there is constipation of the release of these things. And so now we're trying to figure out the physiology, which is not trivial, but we are working on that.
And the other question is, you mentioned in a kind of joking kind of way about the cannabinoids from cannabis, but there's a good case to be made for some kind of study to see whether those cannabinoids from cannabis plant are good inhibitors of the lipoxygenase.
We're going at that a different way. Yeah, so it's a good thing to make a joke about, but I'm not joking about it when it comes to investors. So I have people, which I won't disclose, that are in Colorado. But anyway, that are investing in, but taking a little bit of a different angle. So now that we know, because it's easier to kind of envision telling people they're taking a natural product rather than telling them they're taking something psychotropic. So what we're doing is that we are producing marijuana plants by genetically engineering seeds to produce the components here that we think are going to be important in driving this immunomodulation to immunosuppression. We're getting our first batch. So how we're doing this is we're collecting it in oil. So we're doing an oil emulsion and then seeing what happens. But yes, that's exactly sort of the task that we're taking. Yep.
Fascinating work. I was struck with some background on structural business about the prosthetic group and the anandamide versus the gamma. Was it?
Yes.
The amide group makes a huge difference there, and then it makes a perfect synthetic target. First, to understand what it's interacting with internally and how to make it more or less potent by some sort of synthetic combinatorial chemistry and come up with a much more potent compound, or less potent, as the case may be. But it's fascinating because you can superimpose the two of them, and that amide group makes such a huge difference that it must be a key interaction with the receptor.
Are you proposing a collaboration? Because that sounds fascinating. That takes me out of my realm of expertise. But anyway, all joking aside, if you know anybody who would like to sort of dive down deeper on that, I'm happy to. No, but you make a.
It's just fascinating because I've dealt with things like that in the past, not necessarily to develop it, but we have a couple of projects here in completely different areas where we're looking at that, and the whole idea is being such a small group, that has to be a key interactor with the receptor, and those are not, well, we have to ask for a chemist.
Okay. There's one.
How complicated it would be to change it,
Right.
and then do potency assays like that, and you do have the assay.
Right. That one last question.
In the kind of isolation aspect of what you said, one model that makes me think of, because of what you said, the interaction of the idea and their control immunomodulation with the bottom model you mentioned is the model for necrotized enterocolitis in premature babies, because it's a population that we are routinely using dysbiosis with antibiotics, and later on they
information, sure.
you know, have you any, I heard you mention it's very difficult to isolate the molecule.
No, I mean, so we're getting better at it. You know, I'm not a lipid biochemist by any stretch of the imagination, so my learning curve has been a bit steep, but I collaborate with great people. No, I think that's exactly kind of what we're doing. You know, and I think you sort of start out with your model systems and diseases that you're most comfortable with. Necrotizing enterocolitis, although important, I think in terms of model systems is not very well modeled, right? But certainly we can, you know, Doyle just had a very nice paper published in Cell Press last year looking at the baby microbiome, in particular looking at what happens with these neck babies. So we're not opposed to taking a look at, now we know what we're doing. I mean, it's hard to do work when you have no idea what the hell you're doing. But now we know what we're doing, and I can say that with confidence. And so if you needed to send samples to us, we could, or at least preliminary, do some preps, you know, we could do some assays on that. So.
Very good. With that said, I want to thank everyone for being here today. This is the first of the seminars. As we mentioned, we have tomorrow the workshop in the morning, and then we have a seminar by Dr. Josue. The workshop actually is by Dr. Doyle Ward, that is right there in the corner. He'll be, I think we have like about close to 15, 20 people going to be with him hands-on. And then tomorrow, after that, at 1 o'clock, we have at the Cancer Center, Dr. Josue Perez giving his seminar also. So thank you again. I think there are great ideas here, and this is just the beginning. Hopefully, you know, we'll continue to work together. So thank you very much. Thank you. Thank you. Thank you for everyone that joined online. Thank you for participating in the webinar.